are moving on to our next session of scientific talks by our fellows. The first talk in this session is by Dr. Nitin Gupta, Intermediate Fellow from IIT Kanpur.
Thanks for having me here. Mosquitoes are carrier of deadly diseases like malaria and dengue. Dengue alone kills a person every 25 minutes, and almost 40% of the world population is at risk of catching dengue. And the reason for this wide prevalence of dengue is the carrier of this disease, which is the mosquito Aedes aegypti. It has spread almost throughout the world. The same mosquito also causes chikungunya, yellow fever and Zika. And for this reason, a lot of people are interested in understanding the biology and the behavior of these mosquitoes. And for almost 100 years, people have been trying to understand what attracts mosquitoes to humans. And now we know, based on a lot of behavioral work, that it's a combination of multiple cues. At the short range, it's humidity and body heat that attracts them. But at a long range, it's the body odor and carbon dioxide that we exhale that gives cues to mosquitoes where the human is. Now, these odors are detected by the sensory organs of the mosquitoes, which are mainly the antennae and the maxillary pulps. On these organs, if we look closely, we will find that there are sensory hair called sensilla, and within this sensilla there are olfactory receptor neurons which express olfactory receptors. There are about 100 types of olfactory receptors which work in combination with a protein called orco. And it has been shown in Aedes aegypti that if you mutate orco, then the attraction to humans reduces, which means that the olfactory system is playing an important role. Now, these olfactory receptors, each olfactory receptor detects a particular subset of odors, and based on a lot of work, we now know which receptors detect which odors in many different insects. In Aedes aegypti, a lot of work has been done on specific receptors, and we found people have found the receptors for carbon dioxide and some specific human odors. Now, one thing that people have been trying to do is, given a table like this, that which receptor responds to which odor, if we can figure out what are the similarities among the odors that are responding to the same, that are being detected by the same receptor, we could predict which other odors will also activate the same receptor based on the similarities among those odors. So that has been done, and that works beautifully. But what has not been done is to see, given this data, can we predict the responses of a new receptor by using similarities among the receptors? And the reason this does not work easily is because if you look at the sequence similarities and correlate that with the odor similarity, there was no strong correlation, because very few of the positions in these receptors are actually determining the responses. We were able to overcome this difficulty by identifying which positions in the receptors actually correlate with the responses, and then if we use just those positions, then we can actually predict the responses to a new receptor based on the existing data. So this provides a computational prediction for getting the responses of new receptors, and we show that our predictions work better than a chance prediction. So we tested this method on an independent data set of 26 receptors for about 68 odors, and all these points that are red are those cases where the prediction was better than a chance prediction, whereas the blue points are where the prediction was worse. So mostly red dots indicate that the prediction worked. Now, the receptor neurons send this information to an area of the brain called the antennal lobe. Within the antennal lobe, the receptors of the same type converge into regions called glomeruli, or one glomerulus. Each one is called one glomerulus. And within the antennal lobe, there are two types of neurons: local neurons and projection neurons. Local neurons are local in the antennal lobe, and projection neurons carry information to higher brain areas. And we want to understand how are attractive and repulsive odors represented in the antennal lobe and higher areas to basically understand what is the the separation between attractive and repulsive odors. Now, this data about which odors are attractive and which are repulsive, a lot has been collected over last several decades from various labs. We have also set up a behavioral assay within our lab. The general idea is very simple, that you give one odor from one side in this kind of chamber and release mosquitoes from the center, and from the other side you give a control odor, or typically clean air, and give 5 minutes to the mosquitoes, and if they go towards the odor, then it's attractive. If they go away from the odor, then it's repulsive. So based on, by counting the number of mosquitoes on both sides, we can calculate a preference index, and from that we'll know whether the odor is attractive or repulsive. Now, how is this attraction or repulsion encoded in the brain? So one popular idea that has been suggested from other species experiments is that there may be labeled lines for specific types of odors. So it could be the case that attractive odors are detected by specific receptors, and these receptor neurons project to specific neurons in the antennal lobe, which project to specific areas in the higher brain centers, and separate lines for repulsion. And this seems to be the case for pheromones in Drosophila. The alternative would be a combinatorial code which crosstalks at different intermediate layers. So we wanted to check this in the mosquito Aedes aegypti brain, and for this we are using the technique of patch clamp electrophysiology. So this we set up this technique in the lab. This has never been done before in mosquitoes. So we had to spend quite a bit of time on optimizing the techniques. So the basic setup is like this. So you take a mosquito, immobilize it on a petri dish. It is awake and alive. And then we cut open a small window in the cuticle of the mosquito head, and the brain becomes accessible. So the brain is about 0.2 mm in diameter. And then under high magnification, we put a patch pipette. So this is a glass pipette with a tip of about 1 micron and patch onto a single neuron. Now, we roughly see which area of the brain we are targeting, but it's difficult to identify individual neurons from these kind of images. So we have a tracer dye in the electrode that we inject into the recorded neuron, and then after the experiment we take the brain out, do confocal imaging, and then we can see the morphology of the neuron that was recorded from, and based on that we can identify the neurons post hoc. This is a sample recording from a neuron. So this is a 10-second recording. Odor was given from 2 to 3 seconds, and this neuron showed increase in the number of action potentials or spikes, and we repeat this 10-second trial 6 times. So these are 6 different trials. I'm just showing you the position of the spikes. In every case, it showed a increased spiking rate in response to the odor around this time. So this is one neuron, one odor. So for the same neuron, we gave multiple odor presentations. So this is example from one neuron. For this particular odor, acetonitrile, there was no response in this period, and now we gave different odors. So as we can see, each neuron responds to a subset of odors. The ones that are indicated in red here are repulsive odors to mosquitoes, and the ones marked in green are the attractive odors. And what we found is that the same neuron could respond to both repulsive odors as well as attractive odors. This is one neuron. Now here is another example. This neuron also responded to some repulsive odors and some attractive odors. This is a summary of 43 neurons and 25 odors. So these are 25 odors and 43 neurons, and we clustered the odors according to the response profiles. Ultimately, we found that there is no segregation of attractive and repulsive odors. The same neurons are responding to both, suggesting against the labeled line idea of coding of attractive and aversive odors. So how is the attraction and aversion coded then? Another possibility we considered is that is there a difference in the overall amount of activity that these two types of odors generate on the sensory organs. For that, we use a technique of electroantennogram, in which we basically put a blunt electrode in the antenna directly and measure the overall activity on the organ. And here I am showing you the responses generated on the antenna by different odors. So these green ones are the attractive odors and these are repulsive odors. And again we found that there was no difference in the overall amount of activity. So they are within the same range, attractive and repulsive odors. And the same thing we found also on the maxillary pulp. The attractive odors and repulsive odors and neutral odors were in the same range. So there is no overall difference in the amount of activity generated. Another thing we found is that now we are looking at a single odor here, and these are the responses of different neurons to a single odor, 6-methyl-5-heptene-2-one. So all these neurons are responding to this odor in different animals. But what is interesting is that they are not all responding in the same way. So some neurons respond for show excitatory response for a short while and then show a period of inhibition. Some show a elonged excitatory response, and some show excitatory responses but starting after a delay. And some show excitatory response, inhibitory response, then another excitatory response. So there is a temporal patterning to the responses, and this temporal patterning seems to carry information because it is reproducible or repeatable across trials. So now we are looking more at the temporal patterning and see if the patterning combined with the identity of the neuron carries information about the attractiveness or aversiveness. Another very interesting thing that has been observed in behavioral experiments is that if you look at the response of an individual odor such as lactic acid, it is attractive, and carbon dioxide is also attractive. But if you give both of these odors together, then the behavioral preference to that is significantly enhanced, and it is more than the sum of the individual odors. So there is a synergistic response to the mixture, and this synergism does not seem to occur on the sensory organs because lactic acid is detected only on the maxillary pulps, and carbon dioxide is detected only on the maxillary pulps, and lactic acid is detected on the antenna. It was not very clear in Aedes aegypti whether lactic acid is also detected on the pulp, so we did extracellular recordings from the sensory neurons on the pulp, and we found that it is indeed carbon dioxide is showing a response, but lactic acid did not show a response. So there is no overlap in the sensory organs between these two odors. Also the projections of these from the two organs are in different regions of the antennal lobe. So from the pulp, the projections of the receptor neurons come to 3 specific glomeruli, which are different from the glomeruli of the antennal neurons. So then where does the synergism occur? It could either occur maybe at the level of other neurons in the antennal lobe, or it could occur downstream. So for that we recorded from the antennal lobe projection neurons, and we found that some projection neurons responded only to one of the two odors, but there were some neurons that responded to both the odors. So this neuron responds to lactic acid as well as carbon dioxide and the mixture of both. So there is already crosstalk within the antennal lobe, and this crosstalk can potentially be mediated by local neurons which are densely innervating throughout the antennal lobe. So they are going to different glomeruli, and they are also widely responding to multiple odors. So they are a potential candidate for the crosstalk. And we are also now looking at what type of neurons are these excitatory or inhibitory, and we are finding that they are both excitatory and inhibitory. So this is anti-GABA staining. GABA is one of the common inhibitory neurotransmitters. So this neuron is GABAergic and this one is not. So both excitatory and inhibitory neurons seem to be there. Now another interesting thing is that if we look at these projection neurons, they go to mushroom body and the lateral horn and the higher brain centers. And interestingly, the connections between it is known in many other insects that the connections between projection neurons and the mushroom body neurons are stochastic. So if I look at projections, the connections in one individual mosquito versus another mosquito, they would be different. So this poses a problem for innate preferences, that how are the behaviors consistent from mosquito to mosquito if the neural circuit itself is stochastic in nature. But interestingly, it is also found that even in the neurons that are after this random connection layer, the neural responses are consistent from animal to animal. So the problem becomes, how do we get consistent responses despite the random connectivity? So this we try to understand using computational modeling. So basically we measured the stereotypy using two metrics, correlation based metric comparing the responses across two individuals and for pairs of odors, and another metric that we defined. And with both metrics we found that even though the responses of individual neurons in the second layer were not consistent across animals, the responses of the third order neurons were consistent, and this consistency came from the population of the second order neurons. The whole population response of the second layer was also consistent. Then we looked at the network properties that lead to this consistency. So one thing we found is that the overall size of the network determines the level of stereotypy or consistency that there will be. So larger networks will have more stereotypy, and also the amount of activity in the network leads to more consistency. We also tested this prediction in Drosophila. So in collaboration with Andrew Lane at University of Sheffield, we generated Drosophila flies in which a neuron that limits the amount of activity was mutated or sort of inactivated, and in that case we found that the stereotypy increased. So basically increasing the amount of activity in the network increased the amount of stereotypy, which was consistent with our predictions in the model. And we also found that there is a balance between the amount of randomness and the amount of convergence that happens from the second order to the third order neurons. So if there is more randomness, then more amount of convergence can overcome that to give consistent behavior. So in summary, we have developed a method for prediction of OR responses and shown that it's successful. We have found evidence against label lines for attraction and aversion in mosquito brain. We have shown that attractants and repellents don't differ in the overall amount of activity that they generate on the antenna and maxillary pulps. We have also found that the spike timing seems to carry information about odors. The synergy between carbon dioxide and lactic acid likely originates in the antennal lobe and seems to be could be mediated by local neurons, and this we are probing further. And through computational modeling, we have found various network properties that allow consistent responses from individual to individual. And now we are going to look at see whether the projections of different neurons from the antennal lobe to higher brain areas, are they segregated between attractive or aversive odors, or are they also overlapping? And we are trying to look at the role of the temporal patterning within the olfactory codes. Now in recent years, luckily people have also started developing genetic tools for Aedes aegypti using CRISPR-Cas9. So we are taking advantage of that and also trying to develop transgenic mosquitoes that would express GCAM for calcium imaging or optogenetic proteins for manipulating the activity of neurons in specific areas. With that, I would like to thank my team. A bunch of students have helped, specifically Swikriti, Shifali and Pranjul have collected a lot of the experimental data. I would also like to thank O.P. Singh at the Malaria Research Institute in Delhi for providing the initial batch of Aedes aegypti eggs. Sonal, who is helping us with the transgenic mosquito work. Andrew Lane for doing fly experiments with the in the stereotypy work. Rafi Haddad, who was a collaborator on the OR response prediction work. And Sandeep Verma is my mentor on this project. And the funding agencies including India Alliance mainly, and DBT and UGC for funding. Thank you.
Thank you, Nitin. We have time for maybe one question.
So is the attractive odor is linked with some kind of reward?
So we are looking at innate preferences. So these are odors that they are not trained preferences, but even when mosquitoes are born and they have not been exposed to these odors before, they seem to have these hardwired preferences in the brain, so without giving any additional reward. And these odors that we typically use are components of human odors. So molecules like lactic acid, ammonia, octanol, pentanone, these are all components of human odor, and they are hardwired in their genetics. Thank you.
Thank you. We move on to the next talk now.